good. All right. Here we go. Okay, well, we're looking so far at some of the general aspects of cell signaling and G proteins and both the monomeric and trimeric G proteins and their activation cycles and shutoff cycles. So we went through that. Now we're going to look at several examples of specific signaling pathways. Now once again, those of you who had cell with me, especially if you had it recently and still remember the stuff, there's clearly a huge amount of overlap on this, but we're also going to enter in, but it'll help review and renew your information on these things. And we will also look at some signaling pathways we did not look in cell, especially ones that have special developmental importance. So we're going to start out with two different signaling pathways, and they all use trimeric G proteins. In fact, we call these things the G protein linked receptors. That's what actually, and the signaling pathways based on them. We're going to look at two different kinds. We're going to look at the cyclic nucleotide pathway. We'll say cyclic AMP and cyclic GMP is all very similar. And then we're going to look at the phosphoinositide pathway, which plays a lot of important roles. Okay, now both of these pathways have a couple things in common. One of them is they use exclusively our trimeric G proteins. Second thing, the trimeric G proteins directly bind to the receptor. This is an example of a pathway where the G protein is indirectly activated by a receptor, but never actually touches the receptor itself. Instead, you have several proteins in between that are going to ultimately activate the G protein. That comes later. But in this case, the receptors, at least the occupied receptors, in other words, receptors that absorb light, bound to a ligand, whatever the case is, the occupied receptors directly bind the G proteins. They bind the G proteins and they stimulate that GTP, or rather that GDP for GTP exchange that activates these trimeric G proteins. Then, of course, the alpha subunit falls off. So, in other words, the receptors are classed as guanine nucleotide exchange factors, because guanine nucleotide exchange factors, or GEFs, work by stimulating the G protein to expel its GDP and then pick up a fresh GTP. So since these receptors do that, that means that they're functionally guanine nucleotide exchange factors. So here we have a direct connection between the receptor and the G protein. Now, third thing about this. All of these G protein linked receptors fall into a particular category of proteins because they have structural similarities. In fact, they call them the G protein linked receptor family. Okay, now they have certain features. One of the diagnostic features of these guys, and this is how when people get gene sequences, they run these gene sequences and, of course, calculate out the primary structure amino acid sequence of the protein. They run it through these databases and software and stuff, and what do they find? Well, they find that all of these have certain key features in common. If you have an unknown gene and you get a sequence and run it through the database and it has those features, they'll come back and say, this is another G protein linked receptor. You may not know what it binds or what G proteins it activates, but it's got these similar structural features. So they're all evolved from a common ancestor. Now the cool thing about these, here's the membrane here. These guys are held into the membrane because they have 7 alpha helix regions. Now you might recall from cell biology that an alpha helix, that's a form of secondary structure. It's where you take the amino acid chain and make a spiral coil out of it, and hydrogen bonds within the so-called peptide backbone. In other words, you can ignore the side chains here. Hydrogen bonds stabilize it. So it's a coil. Helix means like a spiral coil, like a spring or something like that. And they have 7 alpha helix regions. They're packed full of hydrophobic amino acids. So of course, those amino acids are going to want to be in a hydrophobic environment. And what's better than that than sitting in a pile of greasy membrane lipids? Okay, so you have an outside portion, and then it snakes into the membrane, 1 alpha helix, and it snakes out, and it snakes back in again, 2, and it snakes back in a 3rd time, and a 4th time, and a 5th time, and a 6th time, and lo and behold, a 7th time, and out it goes. So these guys have these 7 hydrophobic alpha helix regions. And that's what anchor these guys into the membrane. And those, although the amino acid sequences may differ from one of these receptors to the next, they all have these common features. These alpha helix regions are packed full of hydrophobic amino acids. They want to be inside a membrane, and then the rest of the protein sticks outside and inside the cell. And that's going to be divergent. So, each one of these, this is the inside of the cell, each one of these interior portions will bind certain members of the trimeric G protein family and not others. And we mentioned, because we have about 10 genes for each of the 3 subunits, the alpha, beta, and gamma subunits, you have roughly 1,000 potential trimeric G proteins. So that means you can recognize 1,000 different types of receptors. So not surprisingly, these G protein linked receptor pathways are very, very widely used in all kinds of things. But in terms of the final response, these are a couple of major pathways: the production of these cyclic nucleotides and this phosphoinositide pathway, which involves a membrane lipid. Yes?
In this circle that says G protein, what is the last word?
Okay, G protein linked receptors. So in other words, the receptors are connected because they directly bind to G protein. Okay, so all of these G protein linked receptors have that 7 hydrophobic alpha helix regions. That's diagnostic for these proteins, and that's the membrane anchoring region. All right, well, that's the basics about these guys. Now, let's look at the specifics. And you can imagine the variations on the inside and outside. The inside variations from one of these receptors to the next determines what G proteins can bind, what combinations. The outside depends on what ligand can bind, or in some cases like the rhodopsins of our eye, instead of binding in molecule, they absorb light, and that causes the tertiary structure change. Okay, so let's take a look at a couple of these pathways. Now I'm going to do sort of the flow chart type of thing. I heard Dr. Nichols doesn't use flow charts when he covers the same material. Is that true? Did he get that? Some people were telling me he doesn't use it. Can you draw those flow charts again? Okay. Well, anyway, I'm going to do sort of the flow chart. The first one we're going to look at, folks, is going to be the cyclic nucleotide pathway. A little introduction on this. And the quick and dirty involves the production of molecules called cyclic AMP or cyclic, and we represent that as a small c, GMP. Now what are these cyclic nucleotides? These molecules, when they're produced, are going to activate certain families of protein kinases. So here's our amplification step. We produce these nucleotides. Each one of these guys go out, or these cyclic nucleotides. Each one of these guys goes out, binds to a kinase, and turns it on. So here's our amplification. All right, now what's a cyclic nucleotide? All right, let's draw a typical nucleotide. Let's draw AMP, adenosine, that means adenine, mono, single, phosphate. Okay, we're going to have our 5-carbon sugar ribose over here. Connected to this carbon is the base adenine. And then we would have a single phosphate coming off of here. So an oxygen, phosphorus, a double bond, an oxygen, and oxygen. Okay, so that's AMP. Now, what is, and GMP is the same thing. Of course, we just have guanine in there instead of adenine. All right. Now, what is, that's a regular standard nucleotide. Now, what is a cyclic nucleotide? Well, the term cyclic refers to a circle. So somehow we've got to have a circle in this molecule. How are we going to make cyclic AMP? Well, what we're going to do is this. We're going to take this poor phosphate and do something your mama told you never to do to a poor phosphate. We're going to bend and twist this sucker. Okay, so here's the oxygen, phosphorus, the double bond and oxygen here. We're going to take this oxygen here, and we're going to form a covalent bond from this oxygen to the carbon above on the ribose. Question?
I didn't get to draw the regular AMP.
Oh my god.
Oh, okay. Let's get rid of this covalent bond, and here we have it. That's regular AMP. Okay. Now, the thing is, to make cyclic AMP, what we have to do is 2 things. One, we've got to do that condensation reaction, so we make a covalent bond here to this carbon here. That takes energy. Condensation reactions are endergonic. Now, the second thing is covalent bonds have certain angles coming off the atoms. You probably remember from intro chem about bond angles. That's why water, for instance, is like this, because there's these covalent bonds that come off of these angles from the oxygen atom. Okay, now the thing is with these bonds, if you start bending those bond angles, that takes stress. There's only certain limits, like taking somebody's arm and pulling it behind your back. If you pull back a little bit, okay, no problem. If somebody really wrenches it behind your back, you're bending your arm at angles it's not supposed to be, and it causes a lot of pain and it can dislocate things and stuff like that. Covalent bonds are like that too. When you bend that poor phosphate around like that to make a cyclic nucleotide, you're bending those covalent bonds at angles they're really not happy with. That adds additional stress, and it takes additional energy to do so. Just like it takes energy to wrench somebody's arm behind their back, even if they're not fighting back or anything. They generally will, by the way. But if you do that, it takes some energy and effort to do so. Same thing, it takes additional energy, not just to do the condensation reaction, but to bend those covalent bonds, which puts them under stress. So these cyclic nucleotides are molecules under stress. This covalent bond here is relatively weak and easily broken. In fact, you go out and buy this stuff, and you can buy it, it costs a fortune. You darn well better keep it in a freezer, preferably the -80, and take it out only when you're doing experiments. Because even at room temperature, you leave it at room temperature for a couple days, and that bond's going to break, and you're just going to turn your expensive cyclic AMP into less expensive, but still expensive, regular AMP. So the stuff is unstable. That covalent bond can actually break even at room temperature. So we put this molecule under a lot of stress. Once we make it, it's not going to last long, and there's enzymes that will hydrolyze that bond too. Now, how do we make cyclic nucleotides? Obviously, an enzyme has to do it, but the basic thing is, we'll start out taking an ATP, or of course a GTP. An enzyme catalyzes the reaction, hydrolyzes not just the 3rd phosphate, but the 1st, the 2nd, and the 3rd phosphate off. It takes that much energy, the energy of hydrolyzing both the 2nd and the 3rd phosphates off. It takes that much energy to be able to make a cyclic nucleotide. And when you take the 2nd and 3rd phosphates off of ATP or GTP, that gives you about half again as much energy as you do just by taking the 3rd phosphate off. So it gives you additional energy, and you need that. And of course, it's catalyzed by enzymes, so now we get cyclic AMP, and what we call pyrophosphate, which is simply two phosphate ions covalently joined together. And likewise, we do the same thing, get cyclic GMP and pyrophosphate. Now, by the way, pyrophosphate, sometimes you see at the bottom of the ingredient list a number of processed food products. I don't know what they put in there for, by the way, but if you look, sometimes they'll say sodium pyrophosphate or sodium acid pyrophosphate, same thing. That's what it is. It's two phosphate ions covalently joined together. And it's in your food. I don't know what they put in there for. It's harmless, I suppose, but I don't know what it's there for. But anyway, they do put it in as this common additive to commercial food products. Okay, well, what we've got here is how we produce these cyclic nucleotides. Okay, and that has to be catalyzed by enzymes that catalyze the reaction and do the coupled reaction, transferring the energy of the ATP hydrolysis or the GTP hydrolysis to be able to power the making the cyclic nucleotide. Now let's take a look at this particular pathway here. Okay, we're going to start out with the cell membrane. Here's the membrane. Now here's going to be our receptor. And here's the ligand, whatever molecule we're dealing with that receptor binds. You recall, once again, the term ligand refers to a molecule or molecules that bind a particular receptor. It's a generic term like the term substrate, the molecules that bind a particular enzyme. Okay, now, we're going to start it. Here's the receptor. It's already undergone its tertiary structure change because it's got the ligand bound to it. Now, we start out with a trimeric G protein. Alpha, beta, gamma. And it's got GDP in it. Here's our trimeric G protein. Okay, it can now bind to this occupied receptor, so we'll put it in here. And the receptor causes the GDP for GTP exchange. So now GDP goes out, and a GTP comes in. Once that happens, folks, the alpha subunit undergoes a significant tertiary structure change. And what it means is it pops off of the beta and gamma subunit. That causes the beta and gamma subunit to pop off the receptor itself. So now the beta and gamma subunit are off on their own. Here we go. It's right here. Okay, so that separates from the receptor. Now we have our free alpha subunit. I'll represent it as this. And it has GTP. And once again, this tells you it's undergone a tertiary structure change. It can no longer bind to the beta and gamma subunit. Okay, this is the active version of the G protein. Now what it does is it binds to and activates target proteins. And these alpha subunits, what they do is they bind to a large membrane-bound enzyme and activate it. And that enzyme is adenylate or guanylate cyclics. Okay, it activates this enzyme. What this enzyme does is it takes either ATP and makes cyclic AMP, okay, or GTP makes cyclic GMP. The proteins are very similar to each other. The major difference is the substrate is either ATP or the substrate is AMP, but otherwise they're quite similar. So now we start producing our cyclic AMP or cyclic GMP, whichever the case is. We'll just focus on AMP. We make lots of molecules. So what we see here, we see our receptor, we see our intermediary, trimeric G protein, and here's the beginnings of the amplification step. The amplification here is we activate one alpha subunit, activates this enzyme. This enzyme can produce lots of cyclic AMP molecules. And then the cyclic AMP molecules go and bind and activate another membrane-bound protein. And we'll stick the cyclic AMP right here. And these are the protein kinase A's or G's. They're called PKAs or PKGs. They're families, they're diverse families of protein kinases. They're membrane-bound enzymes. And each one of them has different targets when they're activated. There's also other ways of regulating these kinases. But the thing is, all of these kinases absolutely require cyclic AMP or cyclic GMP in order to activate them. There are other kinds of regulatory things that can shut them off even when you got the cyclic nucleotides bound, but in general, they got to be activated by cyclic nucleotides. Once we turn these guys on, then they go and phosphorylate all kinds of things. Some of the things they phosphorylate include membrane-bound transport systems, facilitate diffusion like ion channels, active transport systems. Yes?
Do you have ATP going to CAMP? Is that supposed to be GTP?
Well, or GTP going into cyclic GMP. It just depends. I'm just focusing on this rather than putting both down. Okay. All right, so it could be GTP and cyclic GMP if it's guanylate cyclic. But we're just looking at cyclic AMP. Cyclic GMP does the same thing. Okay, so, and of course cyclic AMP activates the PKAs, cyclic GMP activates the PKGs. Okay, so these guys phosphorylate lots of different things, including things like membrane transport systems, active transport, facilitate diffusion, ion channel, stuff like that. So cells can allow ions in, they can push ions out, they can allow nutrients in, any of a number of different things. Also, we'll say transport. They can phosphorylate and regulate proteins involved in the cytoskeleton, causing things like cell movement, cell shape changes, and the like. Numerous enzymes can be phosphorylated by these guys. That can alter cellular metabolism. In fact, a lot of times we do see changes in cellular metabolism in cells where we activate this pathway. So these things have diverse roles in physiology, development, things like that. All right. Questions on cyclic nucleotide pathways? Now, one example, I'll just give one example. It was used as the cyclic GMP pathway. I don't think they ever got any drugs from this. Some folks that used to be at UAB, they were studying this because it turned out that the cyclic GMP pathway, when it was stimulated, it would cause smooth muscle cell contraction in the walls of blood vessels. And of course, when the walls of blood vessels contract, what happens? Your blood pressure goes up. So one idea, and they're getting drug company money to look at this, is to try to develop inhibitors of this pathway so that you take it and your blood vessel walls couldn't contract as much, meaning you get lower blood pressure. I don't think anything ever came to market from that, but that was some research in that field. So that was one kind of thing. We're affecting the contraction of smooth muscle cells in arteries and veins. Okay, so that's one of our pathways. Now we're going to do a few modifications and make a second pathway here. Make a few changes here. We can use some of this diagram already. Okay, that's a cyclic nucleotide pathway. All right, now why we use one is called the phosphoinositide pathway. Many different roles. Okay, the phosphoinositide pathway involves the cutting in half of a specific membrane phospholipid. Not all phospholipids, just a specific phospholipid. Okay, so we'll say cuts membrane phospholipid and it cuts that phospholipid into two parts. Both parts are regulatory molecules that activate certain aspects of cell function. One of these molecules is is hydrophobic, it stays in the cell membrane, so it affects things in the membrane. The other molecule is quite polar and goes into the cytoplasm and does things there. So one of the cool things about this pathway is we have all bases covered. We've got something that regulates events in the membrane and membrane proteins, and we got something that regulates events in the cytoplasm. Not surprisingly, this pathway is used by a wide range of receptors and a wide range of functions. Now, for instance, if we abolish this pathway completely, you would immediately notice the effects, because you couldn't smell or taste anything. Smell and taste receptors use this pathway. You couldn't see the visual receptors, the rods and cones of the eye use this pathway as well. But it wouldn't bother you because you wouldn't be here, because both the sperm and the egg use this pathway in the response to each other. All the stuff we're seeing in lab about fertilization membranes forming and that type of stuff, the events of fertilization, guess what? This pathway does most of that. And there are other kinds of physiological and developmental events as well. But these are some of the biggest ones. Now, not surprising, many different receptors use this particular pathway. We have a lot of different receptors that will use it. Okay, now let's see how this pathway works. Okay, once again, we have a receptor bound to a ligand. We activate a trimeric G protein. The alpha subunit goes and binds and activates, obviously it's a different alpha subunit than what we had before, but goes and activates an enzyme called phospholipase C, or PLC for short. Okay, a phospholipase is an enzyme that cuts phospholipids. It attacks phospholipids. Okay, let's see what happens here. Well, there are many different kinds of phospholipases. If you want an example of phospholipases, go out and irritate a rattlesnake or a copperhead or a cottonmouth, and you will get phospholipases up close and personal when it bites you. Okay. Because a lot of these, what we call blood toxic or hematotoxic snake venoms, one of the major ingredients in their venom are phospholipases. Because what these enzymes do is they go and chop up membrane phospholipids. What that means is if you get snake bit, at least by a hematotoxic snake, what will happen is that venom starts chewing up the cell membranes around the side of the wound, lysing the cell membranes, causing tissue death and necrosis. Before the nerve cells in those areas die off, they start sending lots of pain signals saying, do something, I'm getting injured. So you get all that pain and stuff. And then if the venom happens to get into your bloodstream, which it generally will, it'll circulate around and start lysing all your red blood cell membranes, spilling out all the hemoglobin, and that's actually the kill mechanism when you get bit by one of these snakes. It lyses so many of your red blood cells that you basically die of real sudden anemia. Of course, for humans, it takes a while. It takes hours for that to happen, even with a rather large and nasty snake. But if you happen to be its typical prey, like a mouse or a small rodent or something, those snakes can take those guys down in seconds. For something like us, it takes much longer. It can occasionally still kill people. How about those snake handlers in the Sand Mountain or whatever? Once in a while, somebody gets fatally bit from those things. Okay, but anyway, so phospholipases cut up phospholipids. Now, most phospholipases are like the kind you find in snake venom. They attack every phospholipid in sight. But phospholipase C is different. It doesn't chew up every phospholipid around. It has one specific phospholipid that it attacks and ignores all the other ones. That phospholipid is only a couple percent of the phospholipids in cell membranes. So if you attack that phospholipid, you're not going to damage the membrane integrity. The membrane is in a semi-liquid state, so it means that you chop that phospholipid up, the other lipids flow in and fill up the gap. Okay, so you're not going to damage the cell membrane with phospholipase C because it has just one specific phospholipid target. So let's take a look at its target. First, let's give the name of this phospholipid. Oops, we can spell it right. Okay, its technical name, phosphatidyl inositol 4,5-biphosphate. PIP2 is a common, there's a couple other abbreviations for it, but PIP2 is a common abbreviation for this phospholipid. Now, what does that mean in ordinary English? Let's go back to our phospholipid structure and see what we have. Typical phospholipid, we've got this glycerol backbone. I'm not going to draw everything in it, we're just going to draw the glycerol. And what we have here, coming off of two of the carbons, are fatty acid chains. Okay, and off of the third carbon is going to be a phosphate ion. That's why we call these guys phospholipids. And then it has to be connected to some polar molecule. Now, in the case of PIP2, that polar molecule is a 6-carbon ring-shaped alcohol called inositol. So it's sort of a hexagon here. A hexagon of carbons, OH groups coming off of all of those. And we happen to also have, lo and behold, we're going to happen to have a phosphate ion on each of two adjacent carbons. That's the 4,5-biphosphate, means two phosphates. Okay, so what we see here, this part of the molecule, like all phospholipids, is very hydrophobic to glycerol and those fatty acids. That makes an extremely hydrophobic molecule. Now, this part is extremely polar, because you've got a phosphate ion, you've got a polar ring-shaped alcohol with all those OH groups, and two additional phosphate ions on it to boot. And one thing that's typical of phospholipids, the hydrophobic fatty acid tails and the polar head group that has a phosphate and something else polar. Okay, now, what does phospholipase C do? Well, it makes a cut. It hydrolyzes a covalent bond. And here is where we do the hydrolysis. We cut the connection between the third carbon and the phosphate and all the other stuff attached to it. And that generates two separate molecules. And the cool thing about this is both of these molecules act as cell regulators. So let's kind of break these guys in two here. This molecule here is called inositol triphosphate because it's got 3 phosphates on it. Sometimes abbreviated as IP3. Now, needless to say, this molecule is polar as all get out. It's got the polar inositol and 3 charged phosphates. So not surprisingly, that molecule, the last place in the world it wants to be is inside a greasy hydrophobic cell membrane. So once it's produced, it gets out of the membrane like a bat out of you-know-where. So that's one thing. Now the second thing we have is our glycerol with 2 fatty acid chains. And we call this diacyl glycerol. Now, by the way, if those of you who remember organic chemistry, an acyl group, you're talking about a hydrocarbon chain, essentially, covalently attached to something. So diacyl, two of these things, that's a fatty acid chain. It's glycerol because it's attached to glycerol. Okay, and we often call this DAG for short. Okay, so what we just done. Now, diacylglycerol is extremely hydrophobic, so it stays inside the membrane. And it too is a cell regulator. Okay, so let's see what we produce and what happens here, what these things do. Okay, so we're going to start out here with, I'll take the phospholipid PIP2, and the enzyme phospholipase C cuts it into diacylglycerol, which we'll put right over here, and IP3, inositol, God, especially when you spell it, inositol triphosphate, which we put right over here. Okay, first, let's see what diacylglycerol does. Diacylglycerol is very hydrophobic. Okay, you guys need a minute, right? She needs a minute. Okay, we're ready, folks? Okay. All right, diacylglycerol, very hydrophobic, it stays in the membrane. What diacylglycerol does is it binds to and activates yet another set of membrane-bound protein kinases. These are the protein kinase C family, or PKC family. And here again, it's another diverse family of protein kinases. Key part about them is they're all activated by diacylglycerol. Many of them can be regulated by other means as well, but diacylglycerol is an activator of all these guys. And once again, when it's activated, it phosphorylates numerous different things. A lot of times it phosphorylates things in the membrane, like involved in membrane transport, opening up ion channels, all kinds of things like that, active transport, facilitate diffusion, what have you. It can also phosphorylate things in the cytoplasm because carbon hangs down the cytoplasm, and that can lead to changes in enzymes. That can also even do things that are going to regulate transcription in some cases. So we can directly or indirectly activate or shut off certain transcription factors. So once again, we can get changes in gene expression from this pathway here. So we could say transport, enzymes, and transcription. Okay, those are examples of things that we activate. Now, what does our IP3 do, or inositol triphosphate? It's very polar, so it goes into the cytoplasm. And what it does is this. There are certain what we call ligand-gated, meaning they're opened or closed by binding some kind of specific molecular ligand. Ligand-gated facilitate diffusion ion channels. And these are calcium ion channels. And you find these guys in places inside, in the membranes of places inside cells that store calcium ions. Okay, so here's, for instance, the endoplasmic reticulum, or ER, especially the smooth ER, has calcium ion concentrations in the millimolar level. Mitochondria stores calcium in there and stuff as well. Now what happens is these things have in their membranes certain types of calcium channels. And IP3 binds to one of the classes of these channels and opens these guys up. And what happens? Recall, inside cells, because calcium ions regulate numerous cellular functions, the cytoplasm of cells have calcium concentrations of less than 1 micromolar. Body fluids, fresh water, salt water generally has millimolar calcium concentrations. So the cells have to actively transport calcium out to keep their internal calcium levels low. But just in case, we also actively transport calcium ions into certain organelles like the ER and stuff like that. They act as calcium storehouses, just in case there's no calcium on the outside. We've got some on the inside in case we need it. So what happens? These guys, these organelles, especially smooth ER, have millimole calcium levels inside, micromolar here. We open these facilitate diffusion channels, calcium ions rush out into the cytoplasm. And calcium ions regulate all kinds of things. There are quite a few proteins, especially in the actin microfilament cytoscale, that are directly activated by calcium ions. There are whole families of enzymes, including protein digesting enzymes, that are calcium activated. Matter of fact, when you try to make cell extracts, one thing you do is throw in chemicals that remove all the calcium. So you shut these proteases down rather than have them chew up your sample. And there's other little tricks too. Okay, we have that. Also, calcium ions activate that small regulatory protein called calmodulin. Calmodulin then goes and binds to and regulates many different target proteins. So calcium ions do all kinds of things in cells. That's why we keep calcium ions out of cells unless we specifically want the stuff in. In fact, many years ago, a drug company developed hopefully a new class of antibiotic. What it did was it was a hydrophobic molecule, slipped into cell membranes, and allowed calcium ions to leak out. Sure enough, it killed bacteria quite effectively. Problem was, it was almost as toxic to eukaryotic cells as it was to bacteria, so they were able to use it as an antibiotic. It's currently used as a research tool. We may or may not, depending on certain features, but we may or may not try to use it in the lab in one of our future experiments. It is kind of fun to play around with this. Now, we see an example of this pathway going because it turns out, among other things, calcium ions stimulate the fusion of storage vesicles with the cell's outer or plasma membrane. In other words, they stimulate secretion. And remember that cortical reaction we keep on seeing in the lab? Those cortical vesicles are stimulated to secrete their contents by calcium ions. And guess where the calcium ions come from in fertilization? It comes from this pathway. When the sperm and egg contact each other, the sperm receptors activate this pathway. They also activate a couple kinases independently, but it activates this pathway. This pathway ultimately leads to calcium release from internal stores, and our friend protein kinase C phosphorylates calcium channels in the membrane so calcium can rush in as well. And that causes the cortical reaction. In other words, the formation of the fertilization membrane, and other things too. Matter of fact, cool little trick. There are certain kinds of dyes that fluoresce, glow in the dark, in the presence of calcium ions. So what you can do is you can introduce these into things like sea urchin or other eggs. Sea urchin eggs work really nicely on that because they're semi-transparent. And then fertilize those eggs. And what you see under a fluorescence microscope is cool. All of a sudden, this stuff is a pretty blue in color. All of a sudden, when the sperm comes into contact with the egg, you see this little bright blue speck. And then it propagates throughout the whole egg in about 30 seconds. Have you ever seen those science fiction films when aliens come up and they fire their photon torpedoes or whatever at a planet, and then you see this huge wave of fire consuming the whole world and destroying it? It's kind of a science fiction thing. Blow up somebody's world, Okay, that's what it looks like under a microscope. It's actually really, really cool to see that. You just see that wave of glowing propagating from the sperm entry site. And if you look at the same thing, you're seeing the fertilization membrane forming, first starting there and then propagating through the whole egg in about 30 seconds. So, that's pretty cool. And of course, this pathway does lots of other things. So what we see here, one reason we use this pathway widely, we've got all bases covered. We've got a regulatory molecule that stimulates a membrane-bound kinase, covers events in the membrane, and some in the cytoplasm. And we got something that does dramatic changes in the cytoplasm by releasing calcium ions. So we can cover all bases. It gets used widely because of that. All right, that Those are two examples, widely used examples, of G protein linked receptors and signaling pathways associated with that. And what that means is, oh my